Hello, class. This is our lecture where we're going to get into dopamine and how it acts in a very specific pathway, our reward pathway. And then we're going to take a look and see how that pathway is affected by addictive drugs. And so first, let's talk about the neurotransmitter and, as we've learned, the neuromodulator itself, dopamine. And then we'll talk about its receptors, how its unique ways that it is transmitted in the brain, and then we'll take a look at the specific areas in the brain that are essential for the reward pathway. And we'll also briefly mention just the other pathways that exist, but we will focus on the reward pathway. Dopamine. So this is a structure that's fairly simple. It has a simple amine group, which is derived from tyrosine, a non-essential amino acid. Right, so this is the amine group. I'm going to do a little color coding here. Right, and this is why amine is in the name dopamine. This also has this structure here, and we call this structure a catechol ring. And so that makes this whole thing, more generally, what we would call a catecholamine. Let me make sure I color code this here. So dopamine is one of the catecholamines. The other two most well-known catecholamines are epinephrine and norepinephrine, or adrenaline and noradrenaline, if you live in the UK. And so this is dopamine, and there are two major subclasses of dopamine receptors. But first, let me just show you this real quick. This is the simplified chemical pathway for making all of the catecholamines. In fact, not just dopamine. And so starting with tyrosine, our non-essential amino acid, creates something called L-DOPA. This is the precursor for dopamine, or neuromodulator of interest now. But from dopamine, you can go on to make norepinephrine and epinephrine. And remember, all of these three combined are the catecholamines: dopamine, norepinephrine, and epinephrine. And L-DOPA is of important therapeutic significance. L-DOPA being I want to say prerequisite, but that's such a silly name, to dopamine. That this is often prescribed for individuals with different conditions that are deficient in dopamine. They will have a prescription to L-DOPA, the precursor to dopamine. Okay, let's move on. And there are two major subclasses of dopamine receptors. In fact, we actually have 5 different major types of dopamine receptors, and they're conveniently labeled just D1 through D5. But they are categorized. D1 and D5 are grouped together, and these are collectively known as the D1 type. I'm going to use DA as a short for dopamine. The D1 type dopamine receptors. And so that leaves our D2, D3, and D4, which are categorized together. And these are known as the D2 type dopamine receptors. And their major functional difference is that, and of course, as we've gone over in our neuromodulation section, as you might guess, our dopamine receptors are metabotropic, which makes them G protein coupled receptors. And we can call these GPCRs, as they often are for shorthand. So none of these receptors are ionotropic. So that means dopamine isn't binding to a receptor and it's not causing an immediate influx of an ion through a channel. It may influence other channels indirectly through metabolic pathways, through the signaling cascade that initiates via these metabotropic receptors. And so what kind of GPCR are D1 receptors, right? They're categorized by the differences in the types of G proteins that are attached to these receptors. And so the D1 type receptors have what we call GS subunits, G protein subunits, GS. And what the GS subunits do is that they stimulate adenylyl cyclase. And the D2 do the opposite. They have G protein subunits that are called GI. And what these do is they inhibit adenylyl cyclase. Okay, I'm missing the adenylyl. There we go. Okay, great. And what is adenylyl cyclase? Well, we mentioned this in one of the neuromodulator examples here, but let me just quickly. Adenylyl cyclases, and I'm going to put cyclases here because there are a number of different types, different classes, and within each class, different types of adenylyl cyclases, but they all do the fundamental same thing. So this is an enzyme, and what it does is, as enzymes tend to do, it catalyzes a very specific reaction, converts ATP into diophosphate and cyclic AMP, a very important signaling secondary messenger. AMP is. I spelled secondary with a number there. Oops. I think you know what I mean. So this is very important in the G protein coupled receptor signaling pathway. And adenylyl cyclases are typically, the vast majority of them are transmembrane. So they are in the membrane. Okay, great. Okay, now I want to talk about how dopaminergic, and let me write this name out. Dopaminergic. How dopaminergic neurons, how they synapse or can synapse in the brain, because there's some extra special ways that we haven't discussed in class yet. In dopaminergic, this simply means having to do with dopamine, or more specifically, having to do with the release or binding or producing of dopamine. Really, it's just sufficient to say just having, relating to, involving dopamine. I don't know why I wrote out that whole list there. So just involving dopamine is a more straightforward and all-encompassing definition. Okay, so we've talked a lot about signal transduction and chemical signaling in the brain. And so here I have a generic neuron here with the dendrites, the nucleus in the middle, and a long axon stretching out to another cell or another neuron. And so most of what we talked about some fairly typical ways for neurons to synapse onto another neuron. And so we talked about there may be a synapse here on the dendrite, right? So synapsing on the dendrite, we may synapse on the axon. If you remember the specific names, we have our axodendritic, axoaxonic, right here, axonic, dendritic. And then we have, and then we will eventually, this will terminate. And we may have a little synapse right here. And this may be axon synaptic. Right. So three really big general ways. Oh, and we have a fourth one, right? That we've discussed, and this is the axon somatic. So I'm skipping the first part. So we have axon dendritic, somatic, axonic, and synaptic. Some general ways for neurons to synapse onto another neuron. But we have two more that I want to talk about that we see in dopaminergic neurons. The first one is called, and I'm probably going to mess up this pronunciation here because I think it's French, I'm just guessing. In passant synapse, and this just means in passing. An in passing synapse. And so let's say we have just a typical textbook synapse here. This is our presynaptic neuron, and let me write it down here. I think it's going to get in the way. Our postsynaptic neuron, and action potentials here cause a release of, fairly mundane neurotransmitter. And we'll just say that it's glutamate. That's going to cause an excitatory postsynaptic potential. Oops, P. Right, so nothing too special here. And so what does our in-passing synapse look like? Well, we have these interesting structures that can exist within an axon. So right here is an axon, and this is the flow of, that was a horrible arrow. The flow of information via action potentials moving down. Right, so this goes up to a cell body, and this goes down to eventual termination. But we can have these bulges here in the axon. And these bulges are called varicosities. This is a varicosity because it's one. So multiple would be varicosities. And what this is a site within the axon of which neurotransmitters can be released. So the axon terminals aren't necessarily the only areas where neurotransmitters can be released. And we see that dopaminergic neurons have these varicosities, right? So these dopamine neurons can be released in passing, right? And there will be a membrane down here. I'll draw the little G part here. That it will bind to, influencing the metabolic processes of this postsynaptic neuron. So that's pretty interesting. These varicosities. And there's one more, fairly simple. Consider this axon terminal. And this is a dopaminergic neuron that releases in response to an action potential. And what we have here in this presynaptic dopaminergic neuron is that it can release onto nothing. So dopamine just gets flooded into the extracellular space, free to sort of diffuse and come in contact with whatever is around in the neighborhood. Sorry, I kind of filled that up with lots of dopamine, didn't I? And so what do we call this? Pretty easy to remember if you remember these four up here: axosomatic, axoaxonic, axodendritic, axosynaptic. We also have extracellular. Extracellular. So, two pretty cool additions to how neurotransmitters can be released. And as we're about ready to see, next section here, we're going to get into the neuroanatomy and talk about those specific areas of exactly where they are in the brain, the dopaminergic pathways. What we're going to find is that unlike neurotransmitters like glutamate or GABA, are major excitatory and inhibitory neurotransmitters, respectively. Dopamine, the neurons that make dopamine are very rare. There are very, very few neurons that produce dopamine, but they extend all throughout the brain. And they release dopamine in the usual ways that you're already familiar with, these neuronal synapses like on dendrites or on the soma. But they also release dopamine neuromodulators in passing and in this extracellular way. And they have axon processes that extend throughout the vast majority of the brain. So a very small number of neurons are controlling the entire dopaminergic system in the brain. And it has great modulatory effects throughout the entire brain. So these small nuclei, dopaminergic nuclei, have an inordinate amount of influence on the whole brain. And as we'll see, dopamine is a pretty profound neuromodulator in how it controls, mediates our behavior. So let's take a look at the neuroanatomy.